invention of genetic code, the credit goes to a scientist called George Gamow. UUA and UAA and UAG are the stop codons. Now here stop codons are nothing but they are present in the termination region. Transfer RNA is needed for reading the genetic code that is present on the mRNA. So translation is nothing but the process of polymerization of amino acids. Hello everyone, a warm welcome to today's session on the chapter 6 of second PUC, that is molecular basis of inheritance. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the process of transcription in DNA. In today's session, we shall discuss about translation, and before moving on to translation, it is very important for us to understand what genetic code is and what are the characteristic features of a genetic code. So to begin with today's session, let us start about studying what is a genetic code. Invention of genetic code, the credit goes to a scientist called George Gamow. So he was actually a physicist who said that genetic code usually is read in 3 words or 3 letters. So if you can see this table here, you can understand that these are nothing but U. It is a nitrogenous base which stands for uracil. So it is always read in the form of 3 codes. Therefore, a genetic code is a triplet codon. And he argued that since there are only 4 bases, that is true, right? What are the 4 bases present in DNA? We have adenine, then there is thymine. Thymine if it is a DNA, or uracil if it is RNA. Then we have guanine, and then cytosine. So here A, this is T bar U, and then we have G and C. So these are the 4 bases. So 4 bases are there. Out of the 4 bases, how can 20 different amino acids be formed? Here, when talking about it, I should say only 4 amino acids can be formed if only one base is considered, right? How can 20 different amino acids be formed? Okay, now let's consider there are 2 bases which are combined. Say for example, AT and TAU. AG, like that. So if such double combinations are made also, 20 different amino acids cannot be formed. So how is it possible for a DNA during the mechanism of translation to produce 20 different types of amino acids coding for a particular protein? Because proteins at the end of the day are nothing but they are long chains of amino acids, right? So that is why. And if they have to code for 20 amino acids, the code should constitute a combination of bases, right? Combination of bases, as you can see here, we have UUU, UUC, UUA, UUG, CUU. Like that, they have made a combination. Imagine if just 2 combinations were there, then also 20 different amino acids were not possible. So that is why they made a combination of the 4 different amino acids that are there, and then they put a table like this, and based on this they found out that because the codons get expressed in the form of a triplet codon, it is possible for the mRNA to synthesize the 20 different amino acids that is needed for the protein synthesis. So that credit actually goes to this particular scientist, George Gamow. So he suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. So now looking onto this table, we can see what they have done is they have taken one nitrogenous base here, and the other nitrogenous base here, and the third one here. So U, U and U. Next we have U, U and C. So that is how they made combinations of it. So based on these combination, if you see here, UUU, it is a triplet codon. It codes for the amino acid phenyl. Likewise, UUC also codes for amino acid phenylalanine or phenylalanine, and UUA codes for the amino acid leucine, UUG also for leucine. Like this, if we count the number of amino acids that are there, so we have amino acid 1 here, the second one, then there is the third, fourth type, fifth, then we have sixth, 7, 8, then 9. Here we have tyrosine, that is 10. Then UUA and UAA and UAG are the stop codons. Now here stop codons are nothing but they are present in the termination region. If you remember, I told you in the previous session of this chapter, transcription begins at a particular region on the DNA, which is called as the promoter region, wherein a specific code is being present, triplet code is being present, and the transcription terminates or comes to an end at a specific region on the DNA where the stop codons are present. So what are the different types of stop codons? So the different types of stop codons, there are 3 different types of stop codons that are present. One is UAA, the other one is UAG, and the third one is UGA. So these are present at the termination region of the DNA strand where the transcription mechanism comes to an end. Now, these stop codons, they do not participate in any of the amino acid synthesis. So therefore, those cannot be considered. Then we have here 11, so 12, then 13, here 14, the 15th type, 16th one, and 17, then we have 18, then 19 and 20. So there are 20 different types of amino acids, or 20 to 21 different types of amino acids that are produced. So this can be possible only by if the codon is made up of 3 nucleotides. So therefore, because the genetic code is made up of 3 codons or 3 nitrogenous bases or nucleotides, they are called as the triplet codon. So this was a very bold proposition because a permutation and combination is involved here, wherein 4 to the power of 3. If you see here, 4 4s are 16, 16 4s are 64. Therefore, totally how many codons are generated? 64 codons are generated. And many more codons are also required for that. But these 64 codons, which appear in the form of triplets, are enough for the synthesis of 20 to 21 types of different amino acids which are needed for the protein synthesis. So that is what they found out. So therefore, any genetic code or any sequence or any gene, if it has to synthesize a particular amino acid, it cannot be in the form of a single nucleotide or a double nucleotide. They should always occur in the form of 3 nucleotides, and therefore that particular gene or genetic code is called as a triplet codon. So this is what we came to know. This particular physicist, he was the one who contributed to all these new discoveries, and that was when people came to know how exactly the protein synthesis occurs, which are nothing but formed by chains of amino acids. And next, Hargobind Khurana, one more scientist, his name is Hargobind Khurana. He was the one who actually synthesized RNA molecules with defined combinations of bases. So he synthesized both homopolymers and copolymers, and there was one more scientist, that is Marshall Nirenberg. So Marshall Nirenberg was one more scientist. He actually gave a particular system which is called as cell-free system for protein synthesis, and this cell-free system for protein synthesis also helped in the cracking of the code. That is how exactly the codes has to be put forward if we need to understand the process of amino acid synthesis. And there is one enzyme which is called as Severo-Ochoa enzyme, and this is nothing but a polynucleotide phosphorylase enzyme And this polynucleotide phosphorylase enzyme was very, very important and helpful in polymerizing RNA with defined sequences in a template independent manner. So therefore, this particular few of the scientists, they found out that this polynucleotide phosphorylase, so this polynucleotide phosphorylase enzyme is very, very important for the enzymatic synthesis of RNA, because after transcription, what happens? During the process of transcription, what happens? mRNA strands get synthesized, right? And if that mRNA strand has to get synthesized, then the polynucleotide phosphorylase is one of the important enzymes that take part in it. And finally, after all these findings, a checkerboard, which I had shown you in the previous slide, it was put for the genetic code, and based on that, the people came to know that the genetic code is a triplet code, and the expression of the genetic code in the form of triplet code, which consists of 3 nucleotides, is very important for the synthesis of a amino acid and therefore for forming proteins. So that is how the genetic code came into existence. Now let's look into what are the salient features of the genetic code. So out of all this which I had told you, what did we finally understand? We finally came to a conclusion that how are the genes expressed? Genes are always expressed in the form of triplet codons such as UAA, UAG, UGA and UCA, UAC, like that. 64 different types of codons it is being expressed. So what are the features of the genetic code? Let us look into. So the codon, as I told you, it is triplet because it consists of one single codon consists of 3 nucleotides in them. So that is why. Therefore, 61 codons code for amino acids. I told you how many codons are there? 64 codons. Will all the 64 codons code for amino acid? No, right? Out of the 64, 61 codons code for amino acid, wherein they produce 20 to 21 different types of amino acids. And what about the other 3? The other 3 are also codons, but the only thing is they do not participate in the process of amino acid synthesis, but instead they act as the stop codon, which helps in bringing about the transcription process to a halt so that next translation step can be carried forward. So therefore, the 3 codons do not code for any amino acid and hence they function as stop codons. They are called as the stop codons. And some amino acids are coded by more than one codon. That is true, right? If you can see here in this checkerboard, one amino acid. Take the example of leucine. Leucine is coded by UUA as well as UUG, CUU, like that many more. So therefore, many number of codons can participate in the synthesis of only one particular amino acid. So that is what that particular point means, that some amino acids are coded by more than one codon and hence the code is degenerate, and the codon is read in mRNA in a continuous fashion. Whenever we read, we don't read it separate, like we don't read it as AUG, UGA. No, it is continuously read throughout the stretch of the DNA. So that is also one of the important characteristic feature of a genetic codon, meaning there are no punctuations involved. Like say for example, I'll write a codon. I'll read it as AUG, right? Will I put a comma here or a full stop here or use any punctuation marks? No, it is always read continuously without any punctuation marks. And the code is nearly universal. Say for example, why universal means if I say UUU codes for phenylalanine, then it will code only for phenylalanine. It will not code for any of the amino acid. So that is why a particular codon is universal to the production of a particular amino acid, or it codes for a particular amino acid. And AUG, as I said, AUG codon has 2 functions. That is, one, it codes for methionine, and also it is the initiator codon. So initiator codon, where is it present? It is present in the promoter region of the DNA strand, which initiates the process of transcription or which starts the process of transcription. This you have studied in the previous session of this chapter. So if you have forgotten, you can just go back and have a look into the video. You will understand. So here AUG is one such codon. So AUG also acts as the start codon, or it also acts as the initiator codon. So it also acts as the initiation codon. And not just that, one more function of the AUG is to produce methionine. And methionine is very, very important. If you go back to the previous session and have a look, you might have observed that in transcription process, the capping of the methionine will occur. That is, at the 5' end of mRNA strand, the capping of methionine has to occur, because the splicing process, if all polyadenylation, if all that has to occur properly during the process of transcription, then the capping of the mRNA with the methionine has to occur. So therefore, AUG is present towards the 5' end of the mRNA strand, and also the DNA, the template, the coding DNA strand, and it helps in the process of initiation of the transcription mechanism. And once transcription is completed and the mRNA strand comes out of the DNA strand, for the mRNA strand, which is a nascent mRNA strand, which is an immature mRNA strand, in the case of eukaryotes, if it has to become a mature mRNA strand, then this particular capping process with the amino acid methionine is very, very important. And that is facilitated by one particular codon, that is AUG codon. Therefore, AUG codon acts as an initiator codon, which initiates or which starts the process of transcription, and also it acts as a codon which codes for the production of the amino acid methionine. And I told you, out of the 64 codons, 61 participates in the process of or 61 participates in the process of or 61 codes for a particular amino acid, and there are 3 codons which do not code for any amino acid, but they are called as the stop codon, because they bring about a halt in the process of transcription, or they terminate the process of transcription. So such codons are UAA, UAG and UGA, and they are called as the stop or the terminator codons. So you can call them as the stop codons, or you can call them as the terminator codons. So these are the salient features of the genetic code. This is a very important question that usually keeps repeating in the exam. Write the salient features of the genetic code for 5 marks. So if you write all these points, 5 marks you will get very easily. So next, let's study about the relationship between mutations and genetic code. So we all know about DNA mutations, right? Sometimes the DNA mutates, it gets altered. So if that alteration occurs, it leads to a lot of disabilities in a body. It leads to a lot, because the protein synthesis will get altered, therefore leading to a lot of disabilities in the body, leading to a lot of deadly diseases and all that. So that is why knowing about mutation is very, very important. There are different types of mutations that occur on the DNA strand or on the genetic code. Let us study about each of these one by one. So the first one is point mutation. So point mutation is nothing but, the name itself suggests point. At one particular point or at one particular nucleotide, the change of the nucleotide will occur. So what happens, we shall look into exactly. So point mutation is a change of single base pair in the gene for beta globin chain that results in the change of amino acid residue glutamate to valine. So here they have taken the example of glutamate to valine. Glutamate is also amino acid. Now I'll just show you glutamate, what is coding for glutamate. Say for example, GAA coding for glutamate, and for valine we have GUU. So GAA and GUU. So moving back further, so what all codes? GAA and GUU. GAA codes for glutamate and GUU codes for valine. So here I have not put the same, because in your NCERT textbook they have given it like this for you to easily understand. But all these are not the nucleotide bases. You all know that because nucleotide bases are always expressed in alphabets A, U, G and C, right? So that is why this cannot be considered, but for you to understand, they have put it like this in the NCERT textbook, so you can understand what exactly the different types of mutations are. So here what happens here is, if it is GAA, then only glutamate will get produced. If it is GUU, then only valine will get produced. Now what happens in the case of point mutation is, it is a change of single base pair in the gene for beta globin chain that results in the change of amino acid residue glutamate to valine. Now here, if anything change occurs, now in the place of GAA, if A gets converted to U, if point mutation occurs and A gets converted to U, glutamate will get converted to valine. But isn't glutamate important? Of course, glutamate is important because it is needed for the production of a particular type of protein. So in that place, if glutamate gets converted into valine because of point mutation, glutamate will not get produced. Instead, in that place, valine will get produced, which will alter the type of protein it produces. Therefore, that protein would have been needed for one of the body function in the human body, right? So that is called as point mutation. Because of the change in one particular nucleotide base, the synthesis of protein will get altered because of the amino acid getting altered, because instead of one amino acid getting produced, in that place another amino acid will get produced. Say for example, instead of glutamate getting produced, valine will get produced, or vice versa it may occur. So that is why. And this results into a diseased condition called as sickle cell anemia. So sickle cell anemia is the best example for valine. How does sickle cell anemia occurs? Sickle cell anemia occurs because of the change of amino acid glutamate to valine. Glutamate will get converted to valine because of the change in one of the nucleotides. So that is why. So here I'll explain to you by taking this. They have given a sentence here, Ram has red cap. So this is one genetic code, because genetic codes are always read in the form of triplet codes, right? So HAS is one genetic code, RED is one genetic code, CAP is one genetic code. Now we shall see that this, say for example, RED is a genetic code that is producing glutamate. It is the one that is producing glutamate. Now what might happen? Can you see here, point mutation has occurred. This remains the same. No mutation has occurred here. This remains the same. No mutation has occurred. But here what has happened here is, one extra nucleotide has got added. Can you see here? B. One extra nucleotide has got added. Now when B got added, can we read it as BRED? No, right? Why can't we read it as BRED? Because genetic code is always read in triplet codes. You cannot read it in 4, right? So that is why BRE will become a genetic code. Now, will BRE produce glutamate? No. Why? Because glutamate was produced by RED. BRE will not produce glutamate. So that is when instead of glutamate, maybe BRE is the one that is producing valine. So therefore, valine will get produced. Now here also DCA became one nucleotide. Here also it changed. Can you see here? So that is nothing but point mutation. Because of the addition of a particular nucleotide or the deletion of a particular nucleotide, there is change in the triplet codon. Therefore, the type of amino acid that gets produced will change, therefore resulting in a diseased condition. That is what happened here. Therefore, what happened? If glutamate, because of point mutation, glutamate gets converted to valine, therefore leading to the disease called as sickle cell anemia. So therefore, in people having this particular disease called as sickle cell anemia, their mRNA strand or the gene present on the mRNA strand will start producing valine instead of glutamate. So that is when the disease will occur. So this is one example for point mutation. Now there is one more mutation which is called as frame shift mutation. Here either insertion or deletion of one or two bases will occur. Therefore, the whole reading of the frame will change. This is called as frame shift mutation. Such mutations are referred to as, if an insertion of a nucleotide base occurs, it is called as frame shift insertion mutation. If the deletion, or if one or two nucleotides get deleted, then it is called as a frame shift deletion mutation. Here also we can explain this by taking this particular example that has been given in your NCERT textbook. So the normal sequence is read like this. RAM, this is one triplet codon, has is one triplet codon, red is one triplet codon, cap is one triplet codon. Now what happened here? Two nucleotides got added. Can you see here? Two nucleotides got added. Now, because two nucleotides got added, what type of mutation is this? This is a frame shift. It is a frame shift insertion mutation. Frame shift insertion mutation. Because two nucleotides got added here. So, now when we read it, what will happen? RAM, it remains as such. Has remains as such, but it will be read as BIR because here what got added here? B and I had got added. So that is why, and it will be read as EDC, then it will be the remaining nucleotide. Now, this was supposed to produce a different type of amino acid. But in this place, can you see what happened here? Entire frame of DNA got shifted because of the addition of two different nucleotide bases there. So that is why it will result in the production of a different type of amino acid. Therefore, the protein will get altered, therefore leading to a number of diseases. Likewise, we have here, say for example, again, three nucleotides got added. What are those three nucleotides? B, I and G. Now, RAM remains the same, has BIG will be read as one, red will remain the same, cap will remain the same. Everything, if you see this normal nucleotide, everything here also remains the same, right? But three different nucleotides got added in between. Now, big will produce one different type of amino acid. Now, these chains of amino acid that will get produced, because of the addition of this, a different protein will get produced there, therefore leading to a lot of diseases. Now, we shall look into deletion. What happens if deletion occurs? So here RAM remains the same, has remains the same. What got deleted here? Just look at this. I'll write the normal nucleotide here. RAM, has, red, cap, right? So here RAM has red cap. Now what happened here? RAM has red cap. R got deleted. Here and all deletion did not take place. Only insertion had taken place. Here what happened? R got deleted, and in the place of R got deleted completely, if you can see, right? So therefore what will remain here? EDC will remain, AP will remain, RAM will remain as such, has will remain as such. That is what has happened here. Therefore altering the amino acid chain, therefore altering the protein, and therefore causing a lot of diseases. Here also, now if I say E has been removed, E is removed, R also got removed, E also got removed. What happened here? RAM will remain the same, has will remain the same, E got removed, therefore R was also removed earlier itself, so therefore it will become DCA. Again different type of nucleotide there, right? Triplet codon there. Again here, I'll remove D. What all got removed there? Deleted R, E, D. All the three are deleted now. What will happen? RAM will remain the same, has and cap. Entire in between one particular triplet codon itself got deleted, right? So therefore what will happen? Chain of amino acid being produced will completely change. Therefore the synthesis of protein will also change. This will result in the diseases in a particular organism. So this is about frame shift mutation, which is of two types: frame shift insertion mutation and frame shift deletion mutation. now we shall look into, because we have understood the genetic code and we understood that if an amino acid has to be formed, then the code should always be in a triplet. And there are 64 codons, out of which 61 participates in the process of amino acid synthesis, wherein few of the codons repeat themselves in synthesizing a particular amino acid, and the other 3 codons, they act as the stop codon, and AUG is the one that acts as the start codon or the initiator codon. All that we understood. Because we understood about that, we shall move into the process of the translation, wherein first we shall study about the tRNA, which acts as an adapter molecule. So in the previous sessions of this chapter, we had studied that 3 types of RNA participate in the process of transcription, translation and all that. They were the mRNA, which is the messenger RNA. Then we have the tRNA, which is the transfer RNA, and we have the rRNA, that is the ribosomal RNA. So first let's study about the tRNA. About the mRNA, in the process of transcription, we studied tRNA and rRNA participates in the process of translation. So before moving on to translation, it is important for us to understand tRNA, which is transfer RNA. So the credit for the discovery of tRNA, it actually goes to Francis Crick. So Francis Crick said that there has to be a mechanism to read the code, right? So in the previous slide, we studied about genetic codes. How will the code get read? Because the codes is in the form of AUG, UAC, UCA, UUU, right? It should be read. How will it be read? So that thought came to Francis Crick, and Francis Crick was one of the contributors to say that the DNA is double helical in nature and all that. We have studied about that previously. So Francis Crick said that there has to be a mechanism to read the code and also to link it to the amino acid, because amino acids have a particular structural speciality to read the code uniquely. So how can that particular code be read? So he postulated, or he stated that there is the need for a presence of an adapter molecule that will actually read the code from one stretch of the mRNA strand to the other stretch. Now, for that particular adapter molecule is called as the tRNA, and later it was also called as the sRNA, which is the soluble RNA. And therefore, however, its role as an adapter molecule was assigned much later. So, what did we understand from this? Overall, after transcription, once the mRNA strands are produced, these mature mRNA strands will have the amino acids in the form of triplet codes, which are nothing but the genetic codes. If these genetic codes are read, then the mRNA alone cannot do it. Therefore, it needs to adopt a particular molecule for it to read the particular genetic code. And what was that adapter molecule the mRNA strand adopts? That is nothing but the tRNA, which is called as the transfer RNA. So therefore, from all this, it is understood that transfer RNA is needed for reading the genetic code that is present on the mRNA. So it will read the code and it will say which particular genetic code produces what type of amino acid. So that is what the credit goes to Francis Crick. So let's study about the tRNA. How does it look? So tRNA, it has an anticodon loop that has bases complementary to the code. So what is there? It has an anticodon loop with bases complementary. That is true, right? If you can see here, this is the mRNA strand. So this is the mRNA strand. On the mRNA strand, there are different codons that are present. AGU, UAC, the 64 codons that I had shown in that checkerboard, right? All those will be present on this mRNA strand. That has to be translated. That has to be read. How will it be read? It will be read by the tRNA. These tRNAs. So, how many tRNAs are there? For a specific code, specific tRNAs are there. Say for example, this tRNA, it has an anticodon. What is an anticodon? So, anticodon means if on the strand AGU is there, anticodon means it will be UCA, right? So, this is, so say for example, this is a codon, anticodon for this is this. And this anticodon, as you can see, it is complementary to the codon, right? It is complementing the codon. Therefore, this particular tRNA will carry the code that is complementing the code that is present on the mRNA strand. So that is how that particular complementary tRNA strand, which is the anticodon, will come and bind with the codon that is complementing itself on the mRNA strand. So that is why they said that the tRNA has an anticodon loop. It is a loop-like structure, right? If you can see, it is a loop-like structure having a 5' end and a 3' end. And in this loop, it is an anticodon loop. Why it is an anticodon loop? Because it has the nucleotide that is complementary to the nucleotide that is present, or the triplet codon that is present on the mRNA strand or on the coding strand or the codon strand. And also apart from that, it has a amino acid acceptor end to which it binds to the amino acid. So if you can see here, the end, it has a amino acid acceptor end at the 3' end. Therefore, what amino acid it is carrying? Serine, right? So therefore, does UCA stand for serine? We shall look into. I'll show you in the genetic code. I'll just go back and show you in the genetic code. It was UCA, right? So UCA, does it? Yeah, can you see here? UCA stands for serine. So that particular tRNA or the transfer RNA carrying the genetic code UCA, which codes for serine, will, moving further, will come and bind here. Now, the AGU code on the mRNA has been read as what? It has been read as serine. So, similarly here also, UAC. So, that anticodon for that tRNA that will come and bind will be U for A for U, and C for G. What does this particular triplet codon code for? It codes for tyrosine. Therefore, it will come and bind. Now, it is understood that UAC codon on the mRNA strand codes for which particular amino acid? Tyrosine. So, that is how they read it. That is why tRNA is a very important RNA that takes part in the translation. They are translating the codes that are present on the mRNA into the amino acid. So that is what they do. So tRNAs are specific. As I told you, they are specific for each amino acid, and for initiation there is another specific tRNA that is referred to as the initiator tRNA, that is in the AUG. AUG is a initiator codon, right? There also there is a particular tRNA that is present, and it is called as the initiator tRNA. And there are no tRNAs for stop codons. Stop codons do not require any tRNA. Now, here is an analysis kind of question. Why does the initiator region require a tRNA? Remember, I told you in the initiator region, there is one codon which is called as AUG, which helps in initiating the process of transcription as well as, or the process of translation, as well as AUG helps in the production of a particular amino acid, methionine. So, if methionine has to be read or translated on the mRNA strand, then a tRNA is required, right? So, that is why it is called as the initiator tRNA. But in stop codons, do stop codons participate in the production of amino acids? No, right? They do not participate in production of any amino acid. What is their function? Their function is to only terminate the process of transcription and translation. So that is why, because they do not produce any amino acids, tRNAs are not needed for the stop codon. So that you should keep in mind. And the secondary structure of tRNA has been depicted that looks like a clover leaf. So the tRNA, if you look at the tRNA, it almost looks like a clover leaf-like structure, and in actual structure the tRNA is a compact molecule which looks like an inverted L. So they can either look like a clover leaf-like structure, or if you look keenly, they can look like an inverted L as well. So that is, so overall we need to understand the importance of tRNA molecule, wherein their main function is to translate the codons that are present on the mRNA strand, therefore translating them to amino acid. So this is the function. So this is about tRNA. Now we have understood about tRNA, we have understood about genetic code. So let us move into studying about what translation is. So translation is nothing but the process of polymerization of amino acids. So far, polymerization of nucleotides had occurred. Now, once the translation mechanism starts, those nucleotides that were formed on the mRNA strand in the form of triplet codons, they should get translated into amino acids. So that particular process of polymerization of amino acids to form a polypeptide is called as translation mechanism. And the order and sequence of amino acids are defined by the sequence of bases in the mRNA that I have shown you previously. So you have the order that is there, as you can see here. Say for example, we have GCA, then we have GUU. tRNA that will come and bind will have CGUY, G complements C, C complements G, and A complements U. And here what comes and binds? That tRNA which has an anticodon carrying the triplet code CAA. Therefore, CGU codes for one particular kind of amino acid, CAA codes for valine. Like this, chains of amino acids will get formed once they start to read from the initiator region up till the terminator region. And the chains of amino acids will finally lead to the production of one kind of protein there which is needed for a specific function in the human body. So the order and sequence of amino acids are defined by the sequence of bases in the mRNA, and the amino acids are joined by a bond which is known as the peptide bond. So you can see here chains of amino acids are formed, right? So they are joined by a bond which is called as peptide bond. Peptide bond. So this is how they start reading. From here itself they start reading. So CUC means it will be, can you just repeat with me? CUC means it will be here G, then here A and G. Now moving back to the triplet codon, let us check what GAG codes for. GAG, which brought the GAG there? The tRNA, right? The particular tRNA, I'll just draw it like this. So tRNA brought the GAG, and this is the tRNA. It brought the GAG and it got binded here. Now we shall look in the genetic code. I'll just go back. GAG. Remember, we are searching for GAG. What GAG codes for? So where is, yeah, GAG. What does it code for? Glutamate, right? Glutamate amino acid. Now can you see here? What is GAG coding for? So here, yeah. So that is how they code for a particular amino acid. Likewise, leucine, then tyrosine, serine. So it keeps on reading like this. Here also one tRNA will come and bind. Here also tRNA comes and bind. Here it will come and bind. Like that it will go on reading. So therefore coding for a particular amino acid. Therefore now as and when it reads, chains of amino acids are produced. Single amino acid is held to one another by the help of a polypeptide bond, forming a polymer of amino acid, a chain of amino acid, which at the end will produce one particular kind of a protein. So that is how the translation mechanism occurs. And the amino acids are joined by a peptide bond, and formation of a peptide bond requires energy, which is obviously provided by the ATP or the adenosine triphosphates. Therefore, in the first phase itself, amino acids are activated in the presence of ATP. Why? Because ATP is needed for the production of polypeptide. So the peptide bond, if the peptide bond gets produced, then only the amino acids will get linked to one another and form a chain of amino acid. And if two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. So the presence of a catalyst will also enhance the production of the ATPs, therefore increasing the production of the formation of, or increasing the rate of peptide bond formation. So here also any catalyst can take part in the formation of a peptide bond because it will increase the rate of the reaction. How? By increasing the rate of ATPs that are produced. Therefore, more number of ATPs get produced, then proper bond is formed between the amino acids. So that is why. So this is about translation mechanism, and the cellular factory responsible for synthesizing proteins is the ribosome. Now from this amino acid, protein has to be formed, and protein has to be formed means what has to take place there? The rRNA, the ribosomal RNA will come to help. That is, the ribosome will come for help. So the ribosome consists of structural RNAs, and it has about 80 different types of proteins it will produce by reading the chains of amino acids. In its inactive state, the ribosome has two subunits, a large subunit and a small subunit. So if you can see here, the ribosome has a large subunit. Now this ribosome is the one which will help in the production of protein out of these chains of amino acids that got formed. So ribosome, how is the structure of the ribosome? It has a large subunit and it has this one smaller subunit. Small subunit. So therefore it is made up of two subunits. So the two subunits always are joined to one another and they go hand in hand in reading the amino acids. When the small subunit encounters an mRNA, the process of translation occurs. If you can see here, it is the smaller subunit that actually binds to the mRNA, and the larger subunit will help in bringing the tRNA to that particular place. So when the small subunit encounters or binds an mRNA, the process of translation will occur, and then that is translation of the amino acid to the proteins will occur. First, what happened? First, the code that is present on the mRNA should be read. So for reading, what will come there? The tRNA will come. So the tRNA will start reading from the start codon. It will start reading, and once it reads, producing complementary codes, it will start producing chains of amino acid. Now that chain of amino acid has to be translated, right? Only if it is translated, then the protein will get produced. So if it has to get translated, then the ribosome should come and bind. And the ribosome has two subunits, one larger subunit and smaller subunit. In that, the smaller subunit is the one which goes and binds to the mRNA, and it will read the particular, and it helps in reading that particular the amino acids that are formed by the tRNA, and it will specify that this is the particular protein that it has to produce. And there are two sites in the larger subunit for subsequent amino acids to bind and thus be close enough to each other for the formation of a peptide bond. As I told you, the larger subunits helps in the participation of the binding of the tRNA, and not just that, it helps in participation of the binding of each of the amino acids with the help of a polypeptide. All that it will help. Apart from that, ATPs are also needed for the proper production of the polypeptide. So this is how the translation mechanism occurs. So whatever codes were there on the mRNA, they will be read by the tRNA. That tRNA will start producing chains of amino acids. Those chains of amino acids that are formed should be translated to the proteins. That translation will be done by the ribosome. So therefore, ribosomes is also one of the important component which plays a very important role in the translation mechanism. So the ribosome, it also acts as a catalyst. Catalyst is the one that increases the speed of reaction. What does it increase here? It increases the rate of translation or production of the proteins. So it also acts as a catalyst, and a translation unit in mRNA is the sequence of RNA that is flanked by start codon and the stop codon. All this I have already told to explain to you. So the translation begins in the start codon and it comes to an end in the stop codon. And I also told you start codon will have a specific tRNA which codes for methionine, whereas the stop codons will not have any specific tRNAs because stop codons do not produce any amino acids. And also mRNA has some additional sequences that are not translated. The entire mRNA strand usually doesn't participate in the process of translation. Some of the sequences are left behind which do not participate in the process of translation, production of amino acid, translation into proteins and all that. And such regions are called as the untranslated regions or the UTR. So the UTRs are present both at the 5' end before the start codon and at the 3' end after the stop codon. That is why, right? Because it is a stretch here. Here also the codons will continue. So, after the stop codon also the amino acids will not get produced. Before the initiator codon also the start codon also the amino acids will not produce. Why? Because the translation mechanism would have occurred only from the start codon up till the termination codon, right? Or the stop codon. So, that regions or sequences of mRNA that do not produce a code for any of the amino acids is called as the untranslated region. So, this is about the translation mechanism, and these untranslated regions are also required for efficient translation process later on. And for initiation, the ribosome binds to the mRNA at the start codon. All this I have explained to you. That is recognized only by the initiator tRNA. What is the initiator tRNA? The initiator tRNA AUG, it will be UAC, right? So, UAC will be the tRNA that is actually will go and bind to the start codon, and the ribosome will further proceed wherein the elongation phase of the protein synthesis will take place, and during this stage complexes composed of an amino acid linked to tRNA will go and bind to the appropriate codon in the mRNA by forming complementary base pairs with the tRNA anticodon. So, I have told you, I have already explained to you, only thing is what is given in the textbook, I am just reading it out. But before itself, I have already told you what happens, how the reading occurs and all that. So, this is how the translation mechanism actually occurs in the case of mRNA strands. So, the ribosome, it moves as and when the codons are needed to be read, the ribosome moves along. It will start moving from the initiator codon up till the stop codon, and the amino acids will get added one by one and it will get translated. It will get translated into polypeptide sequences which is represented on the mRNA, and the end a release factor will bind to it, that is in the stop codon a release factor will bind wherein the ribosomes and all that will get released, the tRNA and all that will get released from that particular region, and only a polypeptide chains of amino acids will be remaining, and that polypeptide chain of amino acid would be for a synthesis of a particular protein. So, that is how translation mechanism occurs. So, this was about this session, entirely understood about the genetic code, the transfer RNA molecule, we understood about the function of the ribosome, and overall we came to know about the mechanism of translation. So, in the next coming session, we shall study about the regulation of gene expression and the human genome project. So, how the genes get expressed, and apart from that there is this human genome project which was carried out for sequencing the complete gene of the human being. So, all this we shall study in the coming session. So, we shall meet again in the coming session. Thank you.